This webinar will introduce you to basic operations on SmartTables, such as creating SmartTables, SmartTables structure, using filters, and performing set operations. To start using SmartTables, select the My SmartTables command from the SmartTables menu. That command takes us to the SmartTables directory, which lists all the SmartTables that are accessible to you. If you are not logged in, you will only be able to see SmartTables that are publicly shared. To create and save SmartTables of your own, and see SmartTables you have saved in the past, you will need to create and log in to a Biopsych account. If you don't have an account, you can click on the Create New Account link, and we'll take you through the process of creating one. You can log into your account up here by clicking on the login link. Once you log in, you will see the My SmartTables tab, which lists all the SmartTables you've created and are private to you. For our first task, we will create a SmartTable of genes from experimental data, explore some of their properties, and look at a method for finding pathways related to those genes. First, we will upload a set of genes from a text file that we've created. We'll take a look at the text file, and this is simply a list of E. coli genes, one per line. To create a new group from this file, we go up to the Operations menu, which lists all the SmartTables commands, and we'll select New SmartTable from Uploaded File. When the dialog window appears, we navigate to our file, then select Open. We'd like the system to analyze the file and try to convert its contents into objects in the knowledge base, and try to make objects should be selected by default. The dialog now asks what type of objects are in the file. Since we are importing a set of genes, we'll select genes. The first line of our file is indeed a column header, so we'll check this box to tell the software not to treat that line as a gene. We now click Upload and Create SmartTable, a process which can take a few minutes based on the size of your file and your internet connection speed. The new SmartTable is now created, based on information from the currently selected organism's knowledge base. Now a few words about SmartTable structure. A SmartTable is very much like a spreadsheet. It has rows and columns. In this case, we have just one column and 121 rows, which correspond to the genes from our uploaded file. Each cell of that column contains a gene object that points back into the knowledge base. We can change the names of the SmartTable by clicking on them to give a better title. Click here to add a description. We can also change the names of the columns by clicking on them. Clicking on a column header changes the actions available for that column, such as the ability to make a new SmartTable or choosing a transformation property. A smart table might have several columns, but the first column will contain the chief objects of interest, in this case genes. But we can create other columns that will hold relationships and properties of the genes. Now we'll add some columns from the Properties menu here. Objects in a knowledge base are associated with a number of properties that can be added to a smart table. This menu shows all the properties that can be applied to a set of genes. Let's add a column for the accession number. And we can add a column for the left end position attribute of genes. We'll sort by the accession numbers to display the genes in that order by clicking a sort arrow on the column. We can also add the nucleotide sequence of the genes as another column via the Properties menu. We can sort by any of these columns. Let's sort by the left end position. Say we are no longer interested in gene GUA-C. To delete this gene from our smart table, select the box next to the row. Then select Delete under the Operations menu, followed by Delete Checked Rows. To add a row, in this case, adding a gene, we'll look up the gene and add it to the smart table. Let's try that, using TRIP-R as an example by using the Quick Search utility. We're taken to the results page for our TRIP-R search. Clicking on the gene takes us to the TRIP-R page, which contains all sorts of information about the gene. Hovering over the Add to Smart Table button will reveal a larger button with a drop-down menu. Clicking on the drop-down menu will let you add the gene to a new smart table or to one of your existing smart tables. Note that the drop-down menu will only display your recently viewed smart tables, not all smart tables you may have. Let's add the gene to our example group by selecting the smart table, then selecting Add. Click on the smart table name to return to the smart table, and the added gene will appear at the bottom of the list. To browse through group members, select Browse from the smart table in the Operations menu. You're now taken to the gene page of the first item in your smart table. Use the next and back links to cycle through the smart table. Click the smart table name to return to the smart table page. Smart tables let you organize and manipulate entries using filters. Filters are powerful functions that allow you to remove existing entities from a smart table based on criteria you select. Or, if you'd like to leave an existing smart table intact, you can use the filter to create a new smart table based on its results. Let's now select the DNA sequence column. Filtering options depend on the type of data in the column. For example, a simple text filter used on the DNA sequence column will display only the genes that contain the substring entered. We'll select the first few nucleotides, copy them, and then select Filter from the Operations menu. We'll paste in our data and change the drop-down menu to show Contains Substring. In this case, we'll let the filter create a new smart table based on the results, so we click Create a new smart table named. Leave the default name below and select Go. We now see that 42 results have been filtered from our initial smart table of 121 genes. We'll now go back to our original smart table. Another way to manipulate smart tables is by using set operations. Set operations are functions that display the union, intersection, or difference between two smart tables. For example, if you had a large group like this one and a small group containing a gene, and you wanted to see which genes from the large group also appeared in the small group, you could use the intersection operation to display all objects in both smart tables. Let's do that now. Then, we'll return to our original smart table. If you had two smart tables you wanted to bring together, you could perform a union using the same menu.